The thyroid gland synthesizes and secretes thyroid hormones, primarily thyroxine, also known as T4, and triiodothyronine, also known as T3, through a series of complex steps involving the uptake of iodine, the production of a precursor protein, and enzymatic reactions. The first step begins with iodine uptake that involves iodide trapping. The thyroid gland actively concentrates iodide from the bloodstream through the sodium-iodide symporter located on the basolateral membrane of thyroid follicular cells. This process is stimulated by thyroid-stimulating hormone from the pituitary gland. Once inside the follicular cell, iodide is transported to the apical membrane, where it enters the colloid, a fluid-filled space within the thyroid follicles. The second step is oxidation and iodination. In the colloid, iodide is oxidized by the enzyme thyroid peroxidase to form iodine. This reaction is facilitated by hydrogen peroxide produced by the follicular cells. Thyroid peroxidase then catalyzes the iodination of tyrosine residues in a glycoprotein called thyroglobulin, which is synthesized by the follicular cells and secreted into the colloid. This leads to the formation of monoiodotyrosine and diiodotyrosine. The third step is coupling of iodotyrosines. The enzyme thyroid peroxidase catalyzes the coupling of iodinated tyrosine residues. Two diiodotyrosine combine to form thyroxine, a hormone with 4 iodine atoms. One monoiodotyrosine and one diiodotyrosine combine to form triiodothyronine, a hormone with 3 iodine atoms. These iodinated thyroid hormones remain attached to the thyroglobulin within the colloid. The iodinated thyroglobulin containing T3 and T4 is stored in the colloid until the hormones are needed. Upon stimulation by TSH, follicular cells engulf thyroglobulin from the colloid via endocytosis. The endocytosed thyroglobulin vesicles fuse with lysosomes in the follicular cells, where proteolytic enzymes break down thyroglobulin, releasing T3, T4, MIT, and DT. The free T3 and T4 are then secreted into the bloodstream. MIT and DIT are deiodinated by the enzyme iodotyrosine deiodinase, and the iodide is recycled for further hormone synthesis. Once released, most T4 and T3 are bound to plasma proteins, mainly thyroxine-binding globulin, and to a lesser extent, albumin and transthyretin. Only a small fraction of thyroid hormones remains free and biologically active. In peripheral tissues, T4 is converted to the more active T3 by the enzyme 5-deiodinase. T3 is the more potent form of thyroid hormone and is responsible for most of the physiological effects. The synthesis and secretion of thyroid hormones are tightly regulated by the hypothalamic-pituitary-thyroid axis. The hypothalamus secretes thyrotropin-releasing hormone, which stimulates the anterior pituitary to release TSH. TSH stimulates iodide uptake, thyroglobulin synthesis, and the release of T3 and T4 from the thyroid gland. The negative feedback mechanism is that the high levels of circulating T3 and T4 inhibit the release of TRH and TSH, maintaining a balance of thyroid hormone production. If you found this video helpful, don't forget to like, share, and subscribe for more informative health content. Stay tuned for our next video.